After read mapping, then we need to assemble these reads into transcripts and estimate the corresponding expression level of each transcript. Cufflink is a tool for transcript assembly and the expression analysis of RNA-seq data. Let's take a look at how cufflinks works. Now suppose we don't know that there are such 3 transcripts structure. We only see these reads. So what should we do next? First, cufflink will look for fragments that are unlikely to appear in the same transcript. For example, here, the red junction read, the line means this read is connected to each other. This is the splice site, and that is a junction read. This yellow junction read and this blue read cannot appear in the same transcript, because if they come from the same transcript, the yellow one should be broken at the corresponding position in the blue, and rather than just directly across. In the same way, the red, yellow, and the blue fragments are mutually incompatible, and the fragments of the same color are mutually compatible. By using each compatible fragment as a node and connecting it with the closest and compatible fragment, we can get an overlay graph. This is the overlay graph. Based on the parsimony principle, cufflinks select a set of paths that are not connected to each other and can cover all reads in the graph as the optimal path, and use it to get the final set of 3 transcripts. In principle, after the assembly of the transcript is complete and the expression level at each exon level is properly processed using certain normalization method, the expression level of the transcript can be directly derived from the expression level of each exon. So here, for the abundance estimation, we can consider RNA-seq is essentially random sampling of transcript sequences. The number of reads mapped to a particular transcript is proportional to its transcript abundance. So we can use the total number of reads that fall on a transcript to estimate its expression. But does it really mean the more reads, absolutely the higher abundance? We have to look at several examples. The first example, we have 2 different genes in one sample. So in one sample means the total read is fixed. The total number of read is the same for gene A and gene B. But the gene lengths are different. Gene A is double the length of gene B. And the transcript abundance, gene A has 2 copies. Gene B also has 2 copies. The mapped read for gene A is 4, and the mapped read for gene B will be 2 if we assume it's a uniform random sampling. And the raw count for the gene A mapped read will be 4, and for gene B will be 2. But does it mean that gene A abundance is twice as gene B? Of course, the conclusion is wrong. The second example, let's look at the same gene, but in 2 different laboratory preparation in 2 different experiments. In the first laboratory preparation, the sequencing depth is pretty high, it's pretty deep sequencing, while in the second one, it's pretty shallow sequencing. The total read in the first experiment is 3 times the total read of the second experiment. Even though the transcript abundance in gene B are same between the 2 experiments, in gene is all 2 copies of the transcript. but the mapped read in the first experiment is 3 times of the mapped read in the second experiment. Is that really means that the gene B abundance is high in experiment 1 but low in experiment 2? Of course, this conclusion is wrong. The third sample example is the 2 samples. We compare 2 samples. Even though these 2 samples have the same laboratory size, means the total reads are same. But in sample 1, the liver sample, the gene A2M only expressed in the liver sample, but not expressed in the brain sample. So the 563 reads used up by this A2M gene in liver sample will be redistributed to all the other genes in brain sample. So even though the read count for everything but the A2M are high in brain sample, however, the only differentially expressed gene is A2M. These 3 examples showed us normalization is extremely important when we do RNA-seq analysis. So when you just want to compare within one sample, but a different gene in one sample, then you can use FPKM as a normalization method. This is fragments per kilobase of transcript per million mapped reads, and is calculated using this formula. C is the total number of mapped reads for a specific transcript. Capital N is the number of total mapped reads, and L is the length of that specific transcript. So FPKM normalized for first the library size, second the transcript length. And the FPKM is used in pair and read. It's called fragments per kilobase of transcript per million mapped reads, while RPKM is used in single and read. It's reads per kilobase of transcript per million mapped reads. And the TPM is transcripts per million. It's another normalization method that is recommended over the FPKM because it can not only compare genes within one sample, but can also compare different genes between different samples. It first adjusts the transcript length, and it also adjusts the total RNA expression. The third normalization method is a method used in the software called RNAseq or RNAseq2. This one is normalized or is considered the different library compositions as I showed in the third normalization example. It uses logs and the median to try to just consider those genes that is normally expressed, moderately expressed in all the samples. So if you want to look at the details of this normalization method, I strongly recommend you to look at Josh Stammer's video in YouTube. So there is another abundance estimation method that is by pseudo-alignment. This one is just developed recently in 2016 and 2017 by 2 software, Callisto and Selma. These 2 software's performance are very similar to each other. They both need reference transcriptome. They do not provide full alignment. That means there's no exact base by base alignment. It's just they roughly estimated this read within those transcript region. Then it will assign this read to this transcript. And it will find all transcript that a read is compatible with, and it's very fast, can map 10 million reads in just a few minutes.